In this module, we will discuss what are the processing level control for the gene expression in the cell. This is a very important segment, that how cell, as a matter of fact, control the expression of the RNA at different level. Now, there are different methods how the cell will control the gene expression, and the one is the alternate splicing. In the alternate splicing, it regulates the gene expression at the level of RNA processing, which provides a mechanism by which a single gene can encode two or more related proteins. You know that the genes are the one which control the regulation or which control the production of the different proteins. But in the alternate splicing, what happens? A single gene, it can encode two or more related proteins in the cells, and the alternate splicing controls that. The proteins which are produced from a given gene by alternate splicing are identified along most of their length which differ in key regions that may affect such important properties like their cellular location, that where they are located, the type of the ligands which they can bind, and the kinetics of their catalytic activity. So in the gene splicing, what happens that the proteins which are produced from a given gene by alternate splicing, they are identical along most of their length. Their length is identical. Their length is the same. But what happens that those proteins differ in the key region. The key region, which are important region, and they affect the important properties of those proteins, like where they will be located in the cells, their type of the ligands which they will bind, and the kinetics which they will show off. Now, alternative splicing, that can become very complex, allowing a wide variety of different combinations of possible exon in the final messenger RNA product. That is their complexity, because their different combination where there are possible exon, which are many in the final messenger RNA products, which are of course the proteins. Just like this, in this diagram, you can see that the alternate splicing of the fibronectin gene, that how it has changed the different spacing and how it is splicing at the different places. Now, another mechanism is RNA editing. And RNA editing is the one in which gene expression that can be regulated at the post-transcriptional level, and that is the post-transcriptional level is by messenger RNA editing, where messenger RNA, as a matter of fact, is edited, and in which splicing nucleotides are converted to other nucleotides after the RNA has been transcribed. So what happens that in the RNA editing, the expression can be regulated at the post-transcriptional level by the messenger RNA, when what happens that the RNA, it has been transcribed, then it can control the RNA editing. Now, RNA editing can create new splice sites, definitely. It can generate stop codons, which are important, or lead to amino acid substitution. So the three things which RNA editing can do is new splice sites creation, it generates stop codons, and at the same time, it can lead to amino acid substitution, various amino acid substitution in the protein which are being produced. Now, why the RNA editing is important? That is, we will show that RNA importance in the editing of the nervous system. Now, in the nervous system, what happened? That RNA editing is particularly important in this system where a significant number of messages appear to have one or more adenine converted to inosines. So, you see, as we discussed previous slide, that in RNA editing, the amino acid substitution is possible. At the same time, the nucleotides have been more edited. So in the nervous system, the adenine, it is converted to inosine, and that is why it's important. Now, the various function of RNA editing has been shown in this transparency. Now, RNA editing, it can be messenger RNA editing, it can be RNA structural, which is there, and the function is that the RNA degradation, RNA replication. That is, it can be either degrade the RNA, destroy it in the parts, or it can substitute the different RNA molecules, and there are the different functions where this RNA editing can perform, which is very clearly illustrated in this diagram. Now, messenger RNA, for example, it can alter the amino acid sequences. It can also alter the future parts of the protein, and it can alter the splicing of the gene. Similarly, in the RNA structure, which differs the difference after the second RNA structure which is there, and it causes a different protein binding or protein binding sites which are there. So how the protein should keep the ligand. Now, this modification in the case of nervous cell, it involves the enzymatic removal of an amino group from the nucleotide. As we have seen, that it removes the amino acid group by an enzyme, and it is subsequently read as G by transcriptional machinery. That when the transcription machinery starts reading, starts producing the new protein, it will read it as a G instead of the other which are there.